Good morning, students. Welcome back to Zoom class for Botany. Today I am going to introduce applications of plant tissue culture. This is going to be very interesting for you. Dear students, first of all, am I audible?
Yes, sir.
Right. Very good. Kindly cooperate until you finish the class, right? Okay. First of all, let me take what is plant tissue culture. Already we have discussed what is plant tissue culture and what are the basic concept of plant tissue culture and what is its techniques and what is its stance we discussed in previous class, right? So let us continue. What is its applications? That is a very interesting area. So first of all, I'm going to introduce applications of plant tissue culture. There are so many applications are there, but we have only few applications are given as a syllabus, right? There are so many points are there because plant tissue culture, which plays very important role in all the fields, right? Like medicine, like agriculture, like pharmaceutical, etc. So the first point is, I'm talking about applications of plant tissue culture, right? As I said, plant tissue culture. A new plant is cultured from a tissue in a synthetic medium under artificial condition, right? That is called plant tissue culture. Simply plant tissue culture is nothing but it is a growing of cell or tissue or organ like root, stem, leaf or calyx, corolla, ambition, dilation, and the endosperm, whatever it may be, or entire plant, which is developed from either from a single cell or from a group of cells in a synthetic medium under controlled condition. That is called as a plant tissue culture. Now we are going to see what is its application. That means what is its uses for mankind, right? Or for agriculture. So first of all, let me take the first point. There is an improved variety. I mean, the somatic hybrids are produced. Somatic hybrids. The somatic hybrids are produced by somatic hybridization. Hybrids are nothing but it is a variety. It can be artificially produced by using somatic hybrid. So, by this technology, we can improve the somatic hybrids and its hybridization process. So that's why the first point is simply improved hybrids can be artificially produced by using somatic hybridization. Then what is somatic hybrid? Somatic hybridization is a kind of plant tissue culture technique by which we can produce hybrid. Particularly, I can say improved hybrids can be produced artificially by somatic hybrid or somatic hybridization. Okay. Second point. Through embryogenesis, we can develop artificial synthetic seed. Artificial synthetic seeds. The artificial synthetic seeds, or it is also known as the seed seeds. These seed seeds or artificial synthetic seeds can be produced more and more by using embryogenesis or somoclonal embryoid. Simply I can say there is a large amount of artificial seeds are produced synthetically or artificially. A number of seeds are produced by somoclonal embryoids or embryoids. Embryoids. No, embryoids are nothing but there is an embryo which is derived from somatic cell that is called somoclonal embryoid or somatic embryoid. Here, the somatic embryoids are involved in the production of artificial synthetic seeds, right? So simply, these synseed or synthetic seeds are encapsulated by using somoclonal embryoid or somatic embryoid. It is a kind of plant tissue culture technique by which we can encapsulate synseeds or artificial synthetic seeds by using somoclonal embryoid or somatic embryoid. Right? Third one, a production of disease-resistant variety. My dear students, third point is a production of disease-resistant variety. Disease-resistant. What do you mean by disease resistant? A variety of plant can be produced by using plant tissue culture technique that shows a resistant power against disease. That is called resistant. Disease resistant variety can be artificially produced by using plant tissue culture technique. I repeat once again, the production of a disease resistant variety in plants can be successfully developed, right? So that is the third point. Then fourth point, production of stress resistant variety. Stress resistant variety. What do you mean by stress resistant? A plant that shows a resistant power against stress. It is of several types. They are salt stress or environmental stress, right? So the stress resistant variety can be artificially produced by using plant tissue culture technique. So such a resistant power can be encapsulated into plants by using one of the plant tissue culture technique, right? So that is the fourth point. And not only this, we can make herbicide tolerant resistant variety. Already we have discussed about Basta, right? So one more point is I'd like to mention here, that is herbicide tolerant variety. Herbicide tolerant. What is herbicide? There is a chemical substances which is used to control herb is called as a herbicide. There is a herbicide tolerant variety can be developed by plant tissue culture technique. Not only that, it is also useful to obtain heat tolerant variety. What is it? Heat tolerant variety. Heat tolerant variety. So it is very interesting area, my dear student. Please understand, right? Next we pass on to the fifth point. The production of secondary metabolites. The production of secondary metabolites. What are metabolites? Actually, metabolites which plays an important role in growth and regulation of plant, right? So, such as alkaloids, steroids, terpenoids, antibiotics, etc. So these are all the secondary metabolites. The secondary metabolites are produced in large amount by using plant tissue culture technique. My dear students, I am discussing about applications of plant tissue culture. This is a content to be expected, very big 5-mark question. Write about the applications of plant tissue culture, right? If it is asked, just start from hybrids, improved hybrids are produced. Second point is artificial synthetic seeds are produced. Then fourth one, it is possible to obtain a disease resistant variety and a stress resistant variety, right? And production of a herbicide tolerant variety, and it is possible to obtain a heat tolerant variety, and it is very much useful in the production of secondary metabolites. Not only that, it is also possible to obtain valuable crops which are cultivated, right, commercially, right? And the tree species in forest itself. Some plants are artificially produced as a craft variety, and the tree species which are naturally grown in forest, the agroforest, right? So this is simply something about applications of plant tissue culture. Sometimes write about the applications of plant tissue culture. If it is asked, you got right these 6 points, right? Very interesting area, my dear student, right? Okay. Students, did you understand? Yes, students, am I audible?
Yes, sir.
Yes, sir.
Could you list out any applications of plant tissue culture that I have discussed with you?
Improved hybrids production.
Very good. Then another students.
Production of disease resistance through
Very
medicine
good.
and
Production
fruitive
of
culture.
disease resistant variety. Then
Production of resistant
Resistant
plants.
plants.
right, very good student. And could you clarify? Could you understand about disease resistant variety? You talk about in few words. Disease resistant. What do you understand about this disease resistant? In few words. Okay, will you list out any applications other than disease resistant and stress resistant?
Production of secondary metabolites.
Secondary metabolites, very good, right? Okay, these are all the applications of plant tissue culture. Dear students, you are kindly requested to pay your attention towards the board, right? You can understand. If you don't understand, at your time ask me, right? I'm ready to clarify. Understand? So try to follow me. Right. So next we pass on to somoclonal variation and the gametoclonal variation. Generally, variation, there is a difference. Variation is nothing but there is a difference in a group of population. It is called a variation. Now, this variation is broadly divided into two types. What are they? The first one is a somoclonal variation. Somoclonal variation. And the second type is a gametoclonal variation. The second type is a gametoclonal variation. I repeat once again, variation is nothing but there is a difference is existed in every generation by means of gamete fusion. The male gamete and the female gametes are fused to form a variation. Now, this variation can be brought about by the fusion of male and the female gamete in plant, right? So, this variation is broadly divided into two types. What are they? The first one is a somoclonal variation, and the second one is a gametoclonal variation. Actually, somoclonal variation is a type of variation. It is found in plants. The plants are regenerated by root, shoot, and leaf, right? So generally, it is found. Generally, it is found in plants. The plants are regenerated. The plants are regenerated by root and shoot. The plants are regenerated by root and stem and leaf, right? That means by using of X plant, which is derived from either from root or from stem or from leaf. The X plant is taken from these organ like a root, a stem, and leaf, by which variation is brought, right, in plant. That is called somoclonal variation. Somo means somatic cell. Dear student, please understand. Somo means somatic cell or body cell of a plant. Here, body cell of the plant. Because the variation is a branch or the body cells of a plant which brings about variation. By regeneration is possible from root or stem or from leaf when we use them as a X plant. So simply, X plant is a part of plant which is used for plant tissue culture technique. The X plant is a small part of plant, or it may be a small piece of plant. It is taken from either from root or stem or leaf. Now, this X plant which brings about variation. These X plant, when it is derived from the body cells or from the somatic cells of an organism, which is regenerated by root and stem and leaf. That is called somoclonal variation. Simply, somoclonal variation is a kind of variation which is found in plant, which brings about by regeneration of root, stem, leaf by plant tissue culture technique. So that is called a somoclonal variation. For that, I'd like to give an example, right? Potato and tomato. This is a potato and tomato are two different varieties. The X plant taken from this potato and tomato, F1 will be tomato. Now that hybrid brings about variation from potato to tomato, right? So dear student, please understand. I'd like to mention this is a potato when it is crossed with the tomato, right? The F1 will be tomato. F1 will be tomato. Now this tomato shows somoclonal variation because the characteristic features of potato and tomato are inherited in F1 generation, I mean in tomato. That shows a somoclonal variation, right? Okay, next we pass on the gametoclonal variation. Actually, gametoclonal variation is possible by the fusion of male and female gamete. It is also found in plant. It is also found in plant, right? It is also found in plant. And this gametoclonal variation, this brings about by the fusion of male gamete and the female gamete, right? By pollen grains and the egg cell. The fusion of male gamete with the female gamete. By the fusion, gametoclonal variation, which brings about in plant, right? So that is the difference between somoclonal variation and the gametoclonal variation. So please understand here, this is a content to be expected in 3-mark question. Write the difference between somoclonal variation and the gametoclonal variation, right? So please understand what is somoclonal variation and what is gametoclonal variation. Simply, I can say the somoclonal variation is a kind of variation that brings about in plants by regeneration of X plant, which is taken from the root, stem, leaf, whereas gametoclonal variation is a kind of variation that brings about in plants by the fusion of male and female gametes. It is naturally occurring by pollination followed by fertilization process. Understand? Okay, I think no more to get confused, right? So next we pass on the micropropagation. Micropropagation. It is very interesting topic. Dear student, please understand micro propagation. Micro propagation. Here micro means a small size. Understand? And propagation means a production. The production of an organism by a small piece of plant, that is an explant, that is called the micro propagation. Right? So micro propagation is a process by which we can produce entire plant when we take explant is in small size explant. As I said already, what is explant? Explant is a part of plant. It may be either taken from a leaf or stem or any other part of the plant. That is called explant, which is used for plant tissue culture technique, right? So I'd like to mention a small portion. This is a leafy portion. I'd like to draw the structure of banana leaf. Dear student, please understand. I'd like to draw the structure of banana leaf. Just imagine. From there, now we are going to use the banana leaf, a small part of banana leaf, a small part of banana leaf, right? Which is cut into a small piece. Now this piece is used as an explant, okay? From the explant, we are going to develop entire banana plant or an individual banana plant is developed from the small explant. The explant is a small part of a plant. It may be taken from a leaf or a stem or a flower or any part of the plant that is used for a propagation process that is called the explant. Now this explant is rapidly propagated by artificial technique or a synthetic medium that is called the micro propagation. So the answer is hidden in the title. Micro propagation means micro means a small size of explant and propagation means a production or producing or developing new banana plant from the small size of explant that is called micro propagation. So that's why, as I said, the micro propagation is a process by which the development of entire plant from the small piece of plant which is used as an explant. This explant may be taken from either from the root or stem or leafy portion or any other part of plant. That is called the micro propagation. So as a result, we can get a new banana plant, right? This is a banana plant. Dear students, you have seen everywhere, right? Banana plant, right? In an artificial condition, under controlled condition, in an artificial synthetic medium, right? Particularly, I can say in laboratory, it is possible to obtain and propagate entire plant. That is called micro propagation. Simply, explant is taken from the leafy portion. What is explant? Explant is nothing but it is a piece of plant pot. It may be taken from either leaf or stem or whatever it may be. From the explant, a small portion of the plant pot that is used for micro propagation. By this technique, we can develop entire plant. That is called micro propagation. So, banana is a right example for micro propagation, right? All these technique has been applicable for only for laboratory in synthetic medium, right? So already we have discussed about that there are so many steps are involved. Culture vessel, culture medium, sterilization. By using a plant tissue culture techniques, we can propagate rapidly in a synthetic medium under controlled condition. That is called micro propagation, right? Hoping that the diagram that is very much useful to understand how this new plant is developed from the small part of it, that is explant, right? So this is one example for micro propagation. And let me give some more example for micro propagation. The first thing is banana. And one more example I'm going to give you, that is pineapple. And one more example I'd like to give, that is strawberry. Strawberry. And one more example I like to give, which is very special, that is a potato. So these plants can be propagated rapidly in laboratory from micro propagation, right? So this is something about micro propagation. Dear students, would you like to give an example for micro propagation? I have given 4 examples. One of you, could you list out?
Pineapple,
Pineapple,
Banana, strawberry,
banana,
potato.
strawberry, and potato. Very good. So, dear student, it is very interesting area, right? Actually, this micro propagation, it is a technique which is used to in order to maintain homogeneity in plants. That is the thing we have to understand, right? In order to maintain standards in industry levels, right? In order to maintain standards or in order to maintain homogeneity. Means the quality of a character is maintained by using of simply micro propagation. So those plants are having good quality in crops, right? They can be artificially produced by this technique. So banana, pineapple, strawberry, and potato, which is very, very, very interesting area, right? So next we pass on to artificial seeds. Another topic I'm going to talk about, that is artificial seeds. As I said repeatedly, artificial seeds are nothing but it is a process by which seeds are synthesized artificially by using plant tissue culture techniques, right? So already we have discussed about the applications of plant tissue culture, right? So artificially, the edible parts of seeds are produced in large quantity. That is called the artificial seed. In order to maintain the homogeneity, right? The quality of seeds, the character, it is to be maintained. So in addition to that, in artificial seeds, right? Now this is a seeds. I like to draw. This is a seeds in which we can develop embryos and endospore and the seed coat, right? This is a, just imagine it is an artificial seed. It is an artificial seed. Now, these artificial seeds are produced by using of some inert material, right? I'd like to mention some inert material. The first one is an agarose. The second one is sodium alginate. What is it? Actually, agarose, it's a kind of polysaccharide which is used for coating of seed coats in artificial seed. Actually, seeds are artificially produced by using of plant tissue culture techniques, right? Particularly, I can say the somoclone embryoids or somatic embryoids. These embryoids are used for making of artificial seeds or synseeds or synthetic seeds. Artificial seed, it is also known as a synthetic seeds. Synthetic seeds. It is also called as a synthetic seeds. The development of synthetic seeds by using plant tissue culture technique, right? Particularly, I can say somoclone embryoids. The embryoids are involved in the production of artificial seeds. Now, this is an artificial seed that contains a rich amount of starch and it also possesses a dense cytoplasm. I'd like to talk about the properties of artificial seed. Now, just imagine this is a seed where you find there is a globus like structure, that is artificial seed. Just imagine in that we find there is a dense cytoplasm, right? There is a dense cytoplasm. What do you understand about a dense cytoplasm? That means the cytoplasm is very thick in nature. It is rich in all the nutrients which are essential for the development of seed development, right? Or for germination process. That means it possesses a vigorous or viability is also retained by the presence of nutrients, right? So it possesses dense cytoplasm. In addition, we can say, right, the presence of starch. Now, starch is a food material that supplies energy for the development of seed when it is shown into the field, right? And one more thing, it also possesses a large amount of protein. And not only that, it is also possessed oil, essential oil, etc. So these are all the properties of artificial seed. I repeat it, so this is a content to expect in board and NEET examination. Dear students, did you understand about artificial seed? My dear students, have you understood about artificial seed? Could you mention the inert material used for seed coating? Dear students, could you mention, students, am I audible?
Yes, sir.
Yes.
Could you mention the inert material used for artificial seed, making artificial seeds?
Agarose.
Agarose, very good.
Sodium.
Sodium,
Agarose and sodium alginate.
right. Definitely the question that is going to be asked, which of the following inert is used for making seed coat in artificial seed? That is a question that you will expect both NEET and board exam, right? Please understand, this is the area to concentrate. One more question, both NEET and board exam. Which of the following inert material used for making seed coat? That is an agarose and sodium alginate. Actually, agarose is a polysaccharide which is obtained from the red seaweeds. I mean, from the red algae. And sodium alginate. Actually, alginate, it is a property of inert material. It is also used in artificial seeds for making seed coat. So that is a difference between agarose and the sodium alginate, right? So both agarose and the sodium alginate, they are essential for absorption of water, and not only that, it also maintains the viability of the seeds in plant, right? So this is very interesting area. Please understand. Write about the inert material which are used for making artificial seed coat in plant tissue culture technique. If it is asked, just write agarose and sodium alginate. Then what are the properties of artificial seeds? So simply it also possesses embryo is that. This portion is the embryo. Yes, student, that you can say embryo, right? And this is a seed coat. It is a seed coat, right? And the endosperm is also present at the center, right? Surely you can say endosperm. So these are all the properties of artificial seeds. Okay, my dear students, next we pass on to what are the advantages of these artificial seeds? What are the advantages? When we use artificial seeds for mankind, what is its advantages, right? That is very interesting topic. Please understand. Follow me. Advantages of artificial seed. What are the advantages? Dear student, it is very important topic and that you can expect in examination. Write about the advantages of artificial seed, right? First of all, let me take the first point. There will be millions of artificial seeds or millions of seeds are produced. Millions of seeds are produced. That means a large amount of seeds are produced by plant tissue culture technique. Right, my dear student, please understand. Second one, it is provides an easy method to obtain genetically engineered product or engineered plant. Right. So it is very easy to obtain genetically engineered plant. Genetically engineered plant with desirable traits or desirable character. Engineered plant with the desirable character. So simply artificial seed that provides an easy way to produce a large amount of genetically engineered plants with the desirable character. Desirable character means what character is essential for insertion of particular gene or in order to bring out particular traits in plant. So it is possible to obtain, to enrich the genetically engineered plants in which we can bring out desirable characters by using artificial seed. Whatever you want, you can insert the gene of such a character that can be inserted into artificial seed, right? That brings about such a traits in plant. So that's why I mentioned it provides an easy way to produce a large amount of genetically engineered plants with a desirable character, right? When we use artificial seed in plant tissue culture, right? And third one, it is very easy to test the genotype of plants. It is very easy to find out the genotype or to test the genotype. To test the genotype. Simply artificial seeds are very easy to test the genotype of plant. The plants are produced by artificial seeds by using artificial seeds, right? So in that plant, we can find out phenotype variation or the genotype variation. Genotype variation. What do you mean by genotype? Actually, genotype is nothing but it is a genetical arrangement, simply the gene arrangement of a character in a plant. That is called the genotype or expression of characters genetically in an organism. That is called the genotype. So simply how the genes are arranged in the chromosome that can be calculated by using artificial seeds in plant tissue culture. That's why as such it is very easy to test the genotype plants. Genotype of plants, right, my dear student? It is very interesting and easy topic. Okay, next one. And the artificial seeds can be potentially stored for a long time. It is possible to store for a long time. For a long time, right? My dear student, please understand. The artificial seeds are produced, as I said, by plant tissue culture technique. Particularly, I can say by using a somocolon embryo, somatic embryo. These seeds are potentially stored for long duration, for many generations. It can be stored potentially. Potentially means the viability, the capacity of seed to germinate in a soil, such a capacity, such a potentiality of artificial seeds can be stored for long duration, for long time, right? So that is also very important advantages of artificial seed, right? The next one is the artificial seeds are producing an identical seeds or identical plant. Artificial seeds are, it is possible to obtain identical plant or identical seeds. Identical seeds or identical plants. So it is very easy to produce a large amount of identical. Identical means though those plants are producing a similar size of seeds, similar morphological characters, they are called identical plants. The plants are produced morphologically similar. They are called identical twins. I think you have heard about identical twins. Both of them are similar in appearance, right? They are called identical twins, right? So like that, here identical seeds, identical plants are produced by artificial seeds. That is also very important. Then the dormancy of the seed can be greatly reduced by using of artificial seeds. Dear student, it is very important area. Please kindly cooperate. The last point is, I think, dormancy of a seed. Dormancy. What do you mean by dormancy? It is a resting period, resting condition of a seed. Before germination, the seed, it is going to be rested. That is called dormancy. The resting periods of a seed, that is called dormancy. Even a single drop of water is fallen in the seed which gets water and starts germination process. In the germination process, first of all, seed coat is broken and followed by liberation of radicle and development from the seeds. So that is called a dormancy. So the dormancy can be greatly reduced. Dormancy can be greatly reduced, right, by using artificial seeds. That's why the span of time or the duration of plant or the total span of a plant can be greatly reduced. Simply, if you take a perennial plant, a biennial plant, a biennial plant means it takes 2 years. In first year, the vegetative growth is completed, and the second year, reproductive phase is to be completed. So that is called a biennial. Right? So the perennial plant simply a plant which completes its span of life within a year. So this 1 year it is to be completed. So the span of life is greatly reduced when we use artificial seeds for agriculture purpose, right? So this is simply something about the advantages of artificial seeds. Right? Almost our time is over, my dear students, right? So here you can expect right about the advantages of artificial seeds. I repeat once again, it is possible to obtain millions of seeds artificially in plant tissue culture techniques. Second one, it provides an easy way to produce genetically engineered plants with a desirable character. And third character is it is very easy to test the phenotypic ratio of a plant, and it is also possible to store potentiality of the seed for long duration. Right, and it is also possible to produce, to obtain a genetically identical plants, or identical seeds or identical plants are produced by this technique. And the dormancy of seeds can be greatly reduced when we use the seeds for germination process. Right. So these are all the advantages of artificial seeds. Write about the advantages of artificial seed or bring out the advantages of artificial seeds. So thank you for joining, my dear students. Almost our time is over. Tomorrow we'll continue virus-free plants, right? I repeat once again. So far, we have discussed about first of all, I'd like to review once again application. There are so many applications that we have discussed. Improved variety, artificial synthetic seeds are produced. Disease-resistant variety can be produced, right? Then herbicide tolerant variety can be developed by this technique, and the heat tolerant variety can be grown by using plant tissue culture technique, and it is possible to obtain a large amount of secondary metabolites like antibiotics, alkaloids, steroids, terpenoids, etc. Right? And it is possible to obtain crops which are cultivated commercially in the field of agriculture, and it is also possible to obtain tree species which are grown for the purpose of medicine, right? And next we discussed variation and types of variation. It is up to they are somatocal variation and gametoclonal variation, right? And next we discussed micropropagation and the types of micropropagation that we have discussed. Banana, pineapple, strawberry, put it there, the example of micropropagation. Then artificial seed. What is artificial seed? And what are the inert material used for making of seed coat? And what is its advantages, right? Dear students, if there is any clarification, any doubt, please ask. Dear student, first of all, did you understand what I taught today?
Yes, sir.
Could you list out the advantages of artificial seeds? Anyone point?
Sir, millions of artificial seeds.
Millions of artificial seeds. Very good, ma'am. Right? Very good.
It is easy to test the genotype of plants.
Very good, ma'am. It is very easy to find out the genotype ratio of a plant. Very good. Then, could
It
you
produces identical plants.
identical plants?
It
Very good.
reduces dormancy.
Very good. The dormancy of seeds can be greatly reduced by using this technique.
Reduced.
Right.
Stored in a long time.
Yeah, it is possible to store long duration. Very good. Very good. Very good. Good, ma'am. Then, could you mention the components of artificial seeds? What do you understand about the cytoplasm of artificial seed? If you take a seed, now this is a seed, right? In which the cytoplasm, what is the nature of cytoplasm? Then what are the materials are present in artificial seeds?
Large nucleus,
Large.
starch grains, proteins, and oils.
Very good, ma'am. Then could you make a typical difference between somatocal variation and the gametoclonal variation?
Somoclonal variation, it is found in plant regeneration. Example, potato cross tomato.
Very good, very good. Then what about the gametoclonal variation?
It is a fusion of it's
Fusion of male and a female gamete. Very good, ma'am. So thank you for joining. Now I'm going to give you a home assignment. Please take down and try to prepare thoroughly, right? Thank you.